Thank you everyone for being here today. I'm really excited for our three speakers in this session. I hope everyone is nice and refreshed after their lunch break and are ready for another 2 sessions of speakers. So first up, we have Dr. Ani Eloyan from Brown University, who will be speaking on the analysis of structural imaging data in cancer. Ani, if you'd like to take the floor.
And thank you so much for organizing the session, and thanks to the conference organizers. I'm sure this was a big task to organize all this online. So let me share the screen. So I will talk a little bit about our team's work on analysis of structural imaging data in cancer, including some of the work of my students, Manhan Hu and Kun Meng. So I will give a little bit of a background first. The work is mainly focused on analyzing cancer tumors, imaging data that shows cancer tumors. These are heterogeneous objects because of their structure, vasculature, cell types within the tumor, and their functional characteristics. So for a long period of time, personalized medicine focused more on the genomic and proteomic technologies and analyzing those for learning about learning some information about the cancer tumors. However, non-invasive medical imaging data are routinely collected for patients with cancer. These images can be used to develop some sensitive measures for understanding cancer tumors in order to be able to predict survival and predict other outcomes of interest. So in this kind of context, we can talk about radiomics. This is a recent area of interest within the field of image analysis, where quantitative features or measures are calculated from tumors or from tumor images that are then used for prediction of diagnosis in cancer. So some of the imaging modalities that have been considered in this context are computed tomography, MRI, PET imaging. So when we talk about radiomics, we are referring to estimation of potentially a very large number of imaging features that are subsequently used for prediction purposes. And the focus being some sort of clinical outcomes of interest. So in this talk, we're mainly focused on survival. The data set that has been used in the studies discussed today is the non-small cell lung cancer data set. This is a publicly shared data set available on the Cancer Imaging Archive, or TCIA. 422 patients are included in this study, and CT scans were available. So about 290 of those were male, and the average age is 68 years. So a little bit of a background on tumor heterogeneity estimation. So biomarkers of tumor heterogeneity can be used for predicting survival. So tumor heterogeneity biomarkers would be our radiomic features. There has been plenty of work in this area, focusing on some main heterogeneity estimation methods. Those include histogram parameters. So that means taking the histogram of the intensities within the tumor and extracting some features of the histogram as measures of tumor heterogeneity. So those could be, for example, the first order statistics or higher order moments. The second kind of major set of features includes shape and size features. So the idea being that tumors that are more smooth or say more circular will have different prognosis than for cancer survival, rather than tumors that have, for example, more non-smooth edges, etc. These also include, of course, volume features. So larger tumors would have different prognosis than smaller tumors. And finally, the texture features or wavelet features that are computed from the intensities within the tumors. So these would characterize the texture of the tumor. So in the first paper that I wanted to discuss, we proposed a method called the Neighborhood-based tumor heterogeneity estimation method. So this is one approach of estimating tumor heterogeneity using a Markov random field-based estimation procedure. So what is the idea behind this approach is the following. So assuming that the tumor is has some collections of cells that have similar features, we can think of the image as potentially having these clusters of intensities that are similar to each other. And the idea is that if the texture is really correlated or associated with the outcome, the survival outcome, then potentially the heterogeneity of these cell distributions within the tumor could be, or intensity distributions within the tumor could be associated with the survival. So how can we estimate these features? So the way that we propose this is by taking these K, assuming that there are K locally homogeneous regions within the tumor. And the idea is to estimate these locally homogeneous regions of intensities. Then at the second step, we calculate descriptive statistics that are based on these regions. So how about the number K? So we can either assume that it is known and calculate, for example, a very large number and see whether that approach would hold here, or we can estimate it by various approaches. For example, by by looking at cross-validation, etc., etc. In order to estimate the cluster membership, so the clusters here would be those homogeneous regions. We use a labeling field, so we define each of the cluster membership for each voxel by lambda v, and we model these using a Markov random field. So the paper has been published in Statistics in Medicine. Everyone is welcome to look at that and let me know if you have any further questions about it. So the main approach here is to estimate the cluster memberships by using a Gaussian mixture model approach. So here we have the distribution for the intensity of the V voxel given by a mixture of Gaussian densities, where the means are and the standard deviations are assumed to be fixed for each mixture element, and we are estimating the weight probabilities. And then to incorporate the spatial structure of the image, as we know, we have this assumption of smoothness, spatial smoothness of the image intensities, which means that if two voxels are neighbors, then their intensities are likely to be closer to each other than for two voxels that are far apart. So that is the smoothness that I'm talking about here when I talk about spatial smoothness. So to incorporate that, we are using a prior distribution for the weight probabilities, pi JV, for each individual voxel. And these are done by incorporating a Gibbs function, through a Gibbs function. So the estimation of the model parameters is performed using an EM algorithm. And again, the details are in the paper. And we have software shared on GitHub for the performing the full analysis described in the paper, including the simulations. So once we can assume that these regions have been estimated using the proposed EM approach, then we calculate the tumor heterogeneity by computing some summary features of these regions. So some of them include cluster size and features. So for example, the average of all of the cluster sizes that were estimated and the standard deviation. So assuming that if for example, the variability of the sizes of these clusters is large, in the image, then it would have a different effect on survival rather than one that has lower variability of the cluster sizes in this setting. Then we calculate some features based on the means, based on the intensities within the clusters, and also the entropy and some other intensity-based features. So we did some simulations to compare the performance of this Markov random field GMM algorithm compared with other approaches, for example, the regular GMM algorithm for estimating the cluster membership, as well as if we were just to use a k-means approach for clustering the intensities without assuming the smoothness of the neighboring voxels. There are some results presented in the paper. One of those is this specific simulation scenario. We have others in the paper, but for the interest of time, I did not include those. And then for prediction of cancer survival, so once we have these features, we use several different methods to predict cancer survival, and we compared the performance of the approach of our proposed approach as compared with some other approaches. So these included if we compute the first order histogram statistics, so if we include only skewedness or only kurtosis, or we include 431 features, including all of these various approaches that I mentioned, or if we use texture features, so some of these include gray level co-occurrence matrix-based features, etc. And then for prediction, we have the Cox proportional hazards model, including PCA for reducing the dimension for some of the features that were estimated, lasso ridge elastic net for regularization, as well as random survival forests, SVM, boosting, and BNN. So we observed that our proposed approach performs as well as or better than other methods depending on the various different combinations of the choice. So these show the cross-validation AUC plots for comparisons. So the approach that discussed, as well as all of these histogram-based approaches, are sort of based on two fundamental assumptions. One is that we have the area of the tumor, or how we would call the mask of the tumor, available before we run the analysis, so that we can extract the tumor intensities only from the image and then perform the analysis based on the tumor intensity values. And then the second is that we are using some kind of information, biological information that, for example, the size of the tumor can have an effect on survival or the texture, etc. So in the second paper, we decided to take a different approach where we are extracting the tumor area located in a box within the image, and we are using the whole box for prediction. So the idea being that at some point this can be used for using the whole image. So for this paper, we decided to use a box around the tumor instead of the whole image, because there is the table, etc., these extra areas that would make it difficult for this initial analysis. So now that, assuming that we have this box around the tumor, we kind of make the hypothesis that perhaps the information within the box can be helpful for, can make the predictions of survival better as compared to just using the tumor image. And then secondly, we are using a convolutional neural network approach to extract a lot more features than those that were discussed before for prediction purposes. So the CNN that was constructed, so this is work led by my former PhD student Manhan Ho, who graduated already. So the CNN approach has the following architecture. So it consists of a convolutional layer, then max pooling, flattening, and then 3 levels of densing, and then the output is the risk of death. from cancer. And so when we ran the analysis first using this approach, we found that the predictions weren't as promising as those that we obtained from the existing methods. Of course, one of the first issues that came to mind is that even though we have hundreds of subjects in this case, it is still quite a small number for this type of analysis when comparing the prediction models using CNN in general. So for that reason, we decided to use transfer learning, which is a method for incorporating existing libraries within the analysis that potentially can improve prediction if the sample size is low. And that was actually quite helpful. So we found that the transfer learning approach using lasso regularized regression for the prediction step resulted in the best prediction, and this was better than the other methods that we looked at. Finally, in the last couple of minutes that I have left, I would like to talk a little bit about another paper that was just published on principal manifold estimation, which is Worklet by my student Kun Meng. So this was his first dissertation paper, where he looked at extending the principal surface approaches that were proposed in the 80s to high dimensional settings. So the idea is that we have high dimensional data that are actually in a low dimensional space, and so we want to estimate the projections of this data within those low dimensional space. So the existing methods for fitting these principal manifolds have several shortcomings. So our proposed approach takes care of the model bias. It improves on computational aspects of the estimation. It improves the sensitivity to outliers, and it can be more easily used in applications. And importantly, this includes a rigorous framework that has a lot of the theoretical results for such a task. So the principal curves, as I mentioned, were developed in the 80s. These were proposed by Hastie and Stoltz in the 80s. So these are sort of like generalizations of the nonlinear regression that we can think about when we think about PCA and its relationship to linear models. Then the principal curves would be the parallel of PCA in terms of its relationships to nonlinear regression. So this is just one preview of the way that we proposed to define the principal manifolds, and there are a lot of details in the paper. Importantly, we propose a model complexity estimation here, where the model complexity refers to the smoothness, so how much smoothness we want to incorporate within our estimate. So I would recommend looking into the paper. In terms of simulations, we compared the proposed method with the Hastian HS method and ISOMAP, that are two of the more popular methods used in this context in terms of estimation of the underlying curves. And we were able to estimate the tumor surface in the same data set. So this is one example of the tumors that I talked about previously. So in summary, we proposed this tumor heterogeneity estimation approach that is based on incorporating neighborhood information. We proposed a method for estimating many radiomic features using CNNs. And then finally, we also proposed the principal manifold estimation approach and used it to estimate the tumor surface by using the vertices provided by a radiologist. So I will stop here, and here are some acknowledgements of my colleagues and students who were involved in this work.
At this point, we will be moving on to our next speaker, who is Dr. Mandy Mejia from Indiana University, who will be speaking on leveraging shared population and spatial information for accurate estimation of subject-level networks.
Okay, thanks so much, Carolyn, and thanks, Ani, for that fantastic talk. So it's a big one to follow. So today I'm going to be talking about leveraging different sources of information in order to accurately estimate brain networks in individual subjects. So here's an overview of the talk that I'll give today. So I first want to start out with just a little bit of motivation for why we should consider going Bayesian for analysis of fMRI data in particular. And then I'll talk about template ICA, which is a framework for using empirical population priors to inform single subject ICA. And then I'll also talk about an extension where we also incorporate spatial priors to improve estimation and increase power. Okay, so first, why should we consider going Bayesian? So I think this question really depends on the goals, and often the goal that I have in mind is understanding the individual. So how can we better understand individual brain organization and connectivity and activation using fMRI data? And this is a tall order in fMRI data because it's very noisy. So historically, fMRI analysis has focused on group level discoveries and group differences. Subject level insights have been challenging to extract accurately due to the low signal to noise in fMRI, but I would argue that they're of vital importance in order to advance fMRI-based research to the next frontiers. And those include biomarker development, longitudinal studies of disease progression and treatment, development and aging, translation to clinical care, certainly, as well as pre-surgical planning. So these are all areas that represent a context where it's very important to extract accurate insights into subject level brain organization. And so why should we go Bayesian? Well, common analysis methods for fMRI treat individuals and brain locations in isolation. So for example, massive univariate task analyses, where we fit a model at every vertex or every voxel separately. I'm showing an example of an estimate of task activation from a massive univariate approach on the right here, and you can see it's very noisy. Another example, though, is also kind of two-level group modeling, where we analyze individuals in isolation and then pull their results together to get a group-level result. So the group-level result might be actually pretty good, but the individual subject-level results don't pull any information from the population. So Bayesian methods, by contrast, leverage information across observations to improve estimation. So here on the right, I'm showing an example of a spatial Bayesian estimate where we're pulling information across the brain, but Bayesian methods can also pull information across populations, which is what I'll be just talking about today primarily. Okay, so if Bayesian methods hold such promise, why are they not more popular and mainstream in fMRI analysis? Well, they present legitimate and major challenges. The first is that you need, kind of high levels of both statistical and neuroimaging expertise to design and implement effective Bayesian models. And so the expertise is key, but also the collaboration is key because few of us have that deep knowledge in both fields. And so it's very important to have meaningful collaborations between statisticians and neuroimagers. And the second is that estimation can be quite computationally intensive. And so there's often a fear of endeavoring along this path because of the computational burden. So I would argue, however, that computational efficiency in Bayesian models is possible. So on the high end of the computational spectrum, we often think about fully Bayesian methods, where we want to fully account for the uncertainty in our priors by assuming hyperpriors on prior parameters and then integrating those out to account for the uncertainty in the form of the prior. And this can indeed be quite computationally intensive in a high dimensional context like neuroimaging. Further down the computational spectrum, or the computational burden spectrum, is empirical-based methods, where we assume the prior is fixed and we estimate the prior parameters during model fitting. But here I'm proposing we go one step further, which is to estimate our priors using external data or a holdout set, and then to just fit our model assuming those priors are fixed. And if we do this, we can make major computational gains because we don't have to worry about actually estimating the prior. And also we can focus, in this case, in the example I'll show here, we can focus on estimating one individual at a time because the prior that's based on the population was estimated a priori. Okay, so hopefully you're motivated to continue following along the talk. So I'll give an example now of how we are taking this approach in the context of single subject ICA. So this is called what we're calling template ICA. So to give a bit of background, single subject ICA aims to decompose, it's a blind source separation algorithm that aims to decompose fMRI data, which I'm showing here as a T by V matrix Y, into a mixing matrix A and a matrix of independent components, which we believe represent brain networks or sources of artifactual origin. And the mixing matrix A is also of interest because that represents the temporal activity associated with each network, and the correlation between those is a measure of the functional connectivity between them. And so people don't actually tend to do single subject ICA in most contexts, and there's a couple of good reasons for that. First is the low signal to noise ratio in fMRI data, which leads to noisy estimates of the independent components and the mixing matrix. But also it's difficult to match independent components across subjects and studies in order to make quantitative comparisons. And this remains true, I would argue, even if we have a lot of data to combat the signal to noise. So instead, traditionally, most analyses will begin with a group ICA and then combine the subject level fMRI data with the group ICs to obtain subject ICs using some method. And there's a variety of methods available to do this. And one benefit of this is that the subject level ICs are matched to the group ICs that we started with. And so if we apply this approach to many subjects, we'll have a set of subject level ICs that are all matched to each other and therefore can be compared. Okay, so template ICA takes this approach in a sense. So it's kind of a hybrid between a hierarchical ICA and one of these two-stage group ICA to subject IC approaches. And so we start with the single subject ICA model, and we just add a second level. And in the second level, we are relating our subject level ICs that we want to estimate to the known group ICs plus an unknown subject level deviation. And so these group ICs, or the population mean, and the variance in those deviations, that's what's known as the template. So the mean and between subject variance of each IC. And we estimate those a priori. And so once we have those estimated, we can focus our model on estimating just a few unknown quantities, including this deviation or the subject level ICs equivalently, the noise variance, and the mixing matrix. And so we take another EM approach, another empirical-based approach, and use expectation maximization to fit this model. And it's very, very fast. So we performed a reliability study to compare this template ICA approach to a standard approach called dual regression, which is very, very popular in practice. And it's kind of comparable in the sense that we can obtain our group level ICs and then relate them to our data to obtain a set of subject level ICs. And so we use data from the Human Connectome project. We use the 25 component group ICA that was provided with the 500 subject HCP release. And we chose 16 ICs from among those 25 that clearly represented recognizable brain networks. And then we used 461 subjects with 2 sessions of data to estimate the template. And that was based on using dual regression to obtain noisy estimates at both sessions, and then to do a variance decomposition to estimate the between subject variance as well as the mean across subjects. And so here's an example of 2 brain networks and their template mean and template variance. So this is the prior mean and variance. So here on the top, I'm showing a default mode network component, and on the bottom, I'm showing an attention network component. And you can see that both are fairly cleanly estimated. And one thing you'll notice is, well, first of all, these means look a lot like the group ICA maps, which we would expect. But also these template variances tend to mimic the template mean in the sense that the areas where the template mean is far from zero are the same areas where we have high between subject variance. And so this is the between subject variance. It does not include the noise variance because we have done that variance decomposition. And so what this is going to provide is strong shrinkage in background areas where we essentially know that all subjects are equal to zero, even though if we get a noisy estimate, it might not equal zero. We know that there's no kind of reliable information across multiple sessions in these kind of background areas. And then in areas where we know subjects tend to deviate, we're going to allow the data to speak more. Okay, so here are some example IC estimates that we get from the Human Connectome project when we apply template ICA, our proposed approach, compared with dual regression. And so you can see that in this, this is relating to this attention network. They have areas of engagement, basically in the areas where we would expect them to have. The template IC estimates are noticeably smoother than the dual regression estimates, but we also get additional pieces of information from template ICA. So one is that we can perform a statistical test to identify areas of significant engagement in an individual subject. So here I'm showing a map of the vertices that exhibit significant engagement after correcting for multiple comparisons. And that's not something that we can get with dual regression. So we also wanted to assess the reliability, how much unique subject level information unique and reliable subject level information are we getting from this approach? So we know they're smoother, but are they actually better? Are they more accurate? And so we applied dual regression and template ICA to data from 20 test subjects with 2 sessions of resting state fMRI using the templates we estimated previously. And then we computed the ICC at every vertex for every IC, every independent component or brain network. And then we computed a summary measure, the I2C2, and we use a weighted version of that in order to prioritize or emphasize the areas of engagement on average across individuals. So the weighted I2C2, just like the ICC, can be thought of as a ratio of variances, where an ICC or an I2C2 of 0 represents the case where we have no reliable subject level information reflected in our estimates, at least not that can be distinguished from the group. And an ICC of 1 means that across multiple measurements, our estimates are exactly the same and that they differ across subjects. Okay, so here on the top, I'm showing 3 different scan durations. So we also wanted to see how our model performed when we had differing amounts of data. So we start out with just 5 minutes of data and we go up to 15 minutes of data. And then I'm showing the weighted I2C2, which is ranging from 0 to 1, using either dual regression or template ICA on the right. And each line represents one of those 16 resting state networks, and they are colored by which overall network they belong to. So as you can see, template ICA does outperform dual regression across all 3 scan durations, across all of the independent components. So this means that not only is template ICA providing smoother, prettier estimates that are that we can do inference on, but also that those estimates are reflecting more unique and reliable subject level information. One thing that we also noticed here is that template ICA quite strongly outperformed dual regression, even based on just 5 minutes of data, where dual regression was based on 15 minutes of data. So even when we triple the amount of data available for dual regression, template ICA still outperforms it. And I think this is a really important point because there's a lot of focus in fMRI research on how much data you need and what scan duration do you need. And I would argue that we always can use more data, but we need to make the most of the data that we have. And if we use better statistical methods, then we can get even more information available from however much data we have. And if that's more data, all the better. Okay, so now in the last few minutes, I'd like to tell you about an extension involving spatial priors. So we take this subject, the same template ICA model, but in this previous model, we assumed that these deviations in this delta matrix were spatially independent. And so here we're going to relax that assumption and assume a covariant structure sigma, where R here represents a spatial correlation matrix, and the D's represent the template variance that we've estimated. And this is going to be representing correlations due to spatial dependence along the cortex in this context. And this is implemented, or this is described in this paper that is currently available on arXiv. So why should we use spatial priors? Well, in kind of more generally, many brain features do exhibit spatial dependence, such as task activation maps. And this is true as we observe in practice of these deviation maps. Even if we don't assume spatial dependence, they tend to exhibit spatial dependence. So we should account for that in our model in order to improve estimation efficiency and power. And so if we use a spatial Bayesian model, we will tend to get smoother, more accurate estimates of these deviations, and also increase power to detect effects. And then when we're doing testing, we can avoid multiple comparisons by taking into account the joint distribution across all locations in the brain. So the EM algorithm in this context is a lot more challenging. It takes about 12 hours for the whole cortex compared with a couple of minutes for template ICA, but it does work. And so if it's worth the extra time, it is available in the template ICAR package. And so what do we tend to gain? Well, we tend to see smoother IC estimates as well as deviations, and we have more power to detect engagements and deviations. Of course, the drawback is the longer computation time, and also it's currently limited to cortical surface analysis. We don't have a volumetric extension at the moment. So here are examples of the independent component estimates we get with this spatial template ICA approach. They're similar to those that we get with template ICA, but slightly smoother. But the bigger difference that we see is in the areas of engagement. So we see the significantly larger areas of engagement because of the increased power with the spatial model. We also, again, can estimate these deviations, this delta term that represents the difference between the individual and the template mean. And here you can notice a bit more smoothness compared with the template ICA model, because this is what the spatial prior is actually being assumed on. And then again, these areas of significant deviation, whether positive or negative, are larger because of the increase in power. And we also did a small reliability study to see if these, again, were more pretty or whether they're actually giving us more information. And what we saw is that if we looked at the scan-rescan correlation across multiple sessions, spatial template ICA, or STICA, was somewhat substantially better than template ICA, but that started to converge as the amount of scan duration increased. And that's kind of a relief, actually, because that means that the computation burden goes up as we have more and more data. So as we get much longer scans, we can perhaps relax the need to incorporate the spatial priors and just use the standard template ICA model. We also look at the scan-rescan overlap of the areas of deviation. And again, at 5-minute scan duration, spatial template ICA strongly outperformed template ICA, whereas at a 15-minute scan duration, they were almost equivalent. But what remains better about spatial template ICA is that we get more power, regardless of the scan duration, at least up to 15 minutes. So I will wrap up. I think I have about a minute left. So to wrap up, template ICA is a framework that uses empirical population priors to inform and improve upon single subject ICA. We've shown that it produces more reliable subject level IC estimates. We can directly estimate what we call subject effects or deviations between the individual brain networks and the population mean. And this is a formal statistical framework that allows for inference to identify significant engagements and deviations. And I should also say that without the spatial priors, it's quite computationally advantageous once you've estimated the prior parameters. And then we've also shown that incorporating spatial priors enhances reliability and power. So I mentioned one extension, spatial template ICA. We're working on a number of additional extensions to this currently. So the first is diagnostic ICA. And so the idea here is that if we had multiple groups with different templates, could we predict the group membership for a new subject based on the compatibility with the group template? And another area is functional connectivity template ICA. And the idea here is that In addition to putting empirical priors on the spatial maps of resting of the brain networks, can we also put priors based on the empirical population, what we observe empirically in the population, on the functional connectivity between the independent components to also improve the estimation of the A matrix, the mixing matrix, which is actually of major scientific importance because of its relationship with functional connectivity. And so just I'd like to briefly point you towards software if you're interested in applying this. Two packages that were used here were the template ICAR package and the SIFTY tools package, which allows for dealing with SIFTY files and GIFTY files and is required for applying template ICA to cortical surface data. And with that, I'd like to thank just a few of my collaborators who contributed to this work. Ying Guo, as you all know from the conference, and thank you so much, Ying, for this fantastic job of organizing this event. Brian Caffo and Mary Beth Nebel at Johns Hopkins, and then my collaborators on the spatial work, Ryan and David. So thank you so much. Also like to thank my funding agency, NIBIB.